we're talking about abiotic signaling. So what that means is, and hopefully you guys, sorry if you hear anything, it's my dog. Hopefully if you guys don't know, abiotic, so bio is life, abiotic is non-living. So these are all going to be non-living signals that get signaled to the plant that are going to lead to a reaction. And so hopefully by the end of tonight, you have a better appreciation of plants as well, because they're truly amazing. And the things that they are capable of doing besides what we talked about last week, like providing us all of our energy from the sun and oxygen, the other things that they're capable of doing are absolutely nuts. They're very advanced. All right. So as a reminder, and this is coming back from when we talked about cell signaling, cells talk to one another, right? And so all the different things that we are going to be talking about today are plant cells talking to one another and sending signals to one another. So that can look a lot of different ways, but usually there's one cell that's sending a signal, one cell that's receiving it, and then we have some sort of response. And those signals and those receivers are going to match up with one another in order to get some sort of response. So not all cells will have that response. It's important to kind of remember that when we're talking about a bunch of the different ways that cells are signaling one another in plants tonight. Another friendly reminder, we have three major categories of signaling. We have direct contact signaling, which is when two cells are touching one another. In fact, you see plant cells right here that can travel or that can pass signals in between the cell walls that are called Plasmodesmata. Then we also have local signaling, which is going to be cells that are close by to one another, that are signaling to one another. And then we have that long distance signaling where we have obviously signals traveling a long distance from target to receptor, and we'll certainly see that today as well. So let's look at some examples. So that's how I'm going to teach most of this. So as I've always said, but if you're just tuning in and you are new to Phytable, you should always have like a pen and pencil, something next to you, so that you can be writing down, or like make a copy of my PowerPoint slide and like start typing comments in for yourself, because there will be like a lot of cell signals and hormones that I'll specifically mention tonight based on the examples that are really important to know about, but might not be explicitly listed for you. Okay? All right. So the very first thing that we're going to talk about, so as I said, I'm going to give you a lot of examples of how plants respond to abiotic signals, right? Just to non-living signals. So we're not going to talk about how plants respond to animals or anything like that, but just different signals that they can get from their environment. So one important thing that I want to make sure that you know about before we jump in, because I'm going to be talking about them a lot with the next two examples, is something called a phytochrome, okay? And a phytochrome is a pigment that exists in plants that is able to detect light. Okay, so my first two examples of how plants respond to abiotic signals is going to be how plants respond to light. So you need to know that there are these pigments, or there are these molecules inside of plants that can actually detect light. Obviously, plants do not have eyes, and yet they know when it is daytime and when it is nighttime, and they know how to respond to different levels of light, and that's going to come into play a lot with germination as well, which is the process of a plant kind of breaking free of its seed and starting to sprout. And so the way that they do that is obviously not through eyes, but through, again, something called phytochromes. And what they actually detect is red light, which is going to be present in high levels during the day. And then they'll also become inactive when they see far red light, which is going to be during nighttime. That's kind of like the light that reaches our Earth. It's called far red light, 730 nanometers. Another reason why detecting red light is so important is because if you remember last week when we were talking about photosynthesis, the photosystems, right, photosystem 2 and photosystem 1, they detect red light. They detect that 660 nanometer light. So it's important that the plant recognizes when that light is readily accessible, and these phytochromes are going to help them to do that. Okay? All right, so super, super important. And it's going to be how we are able to do the next two things, or how plants are able to do the next two things. Okay, so next up, so one and first and the most important, I think, most tested, I guess, and you'll see why at the end of today, but the most tested response that plants have to light is something called phototropism. Okay, so let's talk about a few things here. So trope is going to kind of mean movement, and photo is light always. So photosynthesis, photography, right, involves light. In fact, old photography, you had to have light in order to develop photos. So phototropism strictly means moving towards the light. We're going to talk about gravitropism in a little bit, which would be moving with gravity or against gravity. Okay, but phototropism means moving towards the light. So think to yourself, have you ever seen a plant climbing up a building, right? Like ivy or something like that. I'm sure you all have, right? So if you're thinking about that, or you're thinking about how you might have a house plant that tends to grow towards a window, what you're seeing there is phototropism. Plants will move towards light. And so what we can see here is kind of how scientists deduced where that signal was being detected and what kind of signal it was. And we're going to see that on the next page, okay? So we have to go back to a time where, like, scientists see, yes, plants are moving towards the light, right? And obviously you might be thinking to yourself, well, that's really beneficial to the plant, right? Obviously plants should want to move towards light because that's where they're getting photons, that's where they're getting their energy source, right? But how and why? Well, how mainly are they moving towards the light, okay? So Darwin set up an experiment a long, long time ago to kind of figure out, okay, where is this signal being received by the plant? Is it being received throughout the whole plant? Is it at the top of the plant? Does it have to be directly contacting the plant? What's going on here? And so they set up an experiment, and this is what it looked like. They had 4 different groups. They had one group which had a little seedling that had the top removed. They had one group that had the top covered by an opaque, that means like non-see-through, so something that's like thick, like hard plastic. That would be like not a color that's clear. I'm literally stumbling over words right now, but hopefully you understand what I mean. They had one plant that was covered with a translucent or see-through cap, and they had one plant that was covered at the base with a cap that was opaque, right? That was not able to be seen through. Okay, and basically what they saw was the obviously, as you can see from the diagram, the plant that was covered with the translucent or see-through cap and the plant that had its base covered are fully capable of phototropism. They're fully capable of moving towards the light. Whereas the plant that had the top removed, or the plant that had the cap that was not see-through, that light could not get through, they are not phototroping. They're not moving towards the light. And so you see they're still growing straight up. Okay, and what this told scientists at the time was that the cells that were creating this reaction are being triggered at the top of the plant, right? And so if the top of the plant can see or detect light, those phytochromes, right, are at the top of the plant, then this reaction will occur. If those cells are removed, or if those cells are unable to detect the light because they have an opaque cap over them, then this plant will not phototrope. This plant will not move towards the light, and we will not see this reaction occur. Okay, so that was step 1. Those were the results that they saw. They're like, oh, this is great. We figured out that this reaction is happening in the top of the plant. Okay, and again, this is an abiotic signal that it's responding to, and we're trying to figure out why. Okay, now, next step. So we figured out where the signal is being detected. Now we want to figure out how that signal is moving. Okay, so think about it. We have a couple options, right? Cells may be signaling to one another with direct contact. It might be a local signal. It might be a long distance signal, or some combination perhaps. But there's a kind of an easy way to find out. So if cells are signaling to each other locally or directly, right, with direct contact, then they should be able to signal to each other no matter what, right? So basically this is what scientists did. So Boysen and Jensen, they decided that they could determine what kind of signal this was by basically putting a barrier in between the top of the plant, which we now know is the thing that's producing the signal, and the bottom of the plant. Okay, so hear me out, and I promise I'll explain why this is super important. Okay, so what they did is this. They cut off the top of the plant, and they connected it to the bottom of the plant with something like gelatin or agar. Like, think of like a jello material, something that's squishy. It's permeable. Permeable means things can get through it. Okay, so we have the top of the plant, something that's permeable that things can get through, and the bottom of the plant. And then they do another experiment where they have the top of the plant and something called mica, which is basically a type of rock, and the bottom of the plant. Signals can't get through the rock. Signals can get through the agar. So you're like, okay, great. So this is a pretty easy experiment then. And obviously we see the permeable situation on the left is phototroping, and the impermeable situation on the right is not. Why does this matter? What does this tell us? Why are you listening to me talk right now? All great questions. Basically, what this told scientists was, no, this is not necessarily a direct contact or local signal. If it was, right, then we would still see phototropism over here, at least in the top of the plant, because it's detecting the signal and it's sending the signal to the cells around it, and we would see some sort of phototropism. Because we don't see this, we know that phototropism is caused by a hormone or a long-distance signal, okay, because it is originating up here, but it has to be able to travel to other parts of the plant in order for phototropism to occur. So the way that I like to think of it, right, hormones are long-distance messengers. We got to get from point A to point B, okay? If we have a giant rock in our way, we can't get the message to point B, and nothing will happen. All the things over here that need to hear that signal will not get it because, ooh, we're getting stuck against a rock, okay? Now, if it's a long distance signal and we have something permeable in between, okay, then we can get from point A to point B and we will see phototropism to occur, okay? This would not matter in the slightest if this was not a long distance hormone, right? Because it would have no distance to travel and it would therefore not be significant that something was blocking it from the rest of the plant, okay? But because this is significant, because we don't see phototropism occurring on the right, scientists are like, oh, this signal has to travel a long distance then, because when it can't travel a long distance, nothing happens, okay? So they're like, oh, this is a hormone that is causing this reaction. Specifically, the hormone is, oops, sorry, I have a couple questions. Okay. So let me start answering Vera because that's the slide that I'm on right now. Or, well, they're all Vera's questions, but I'll answer all of your questions, Vera, I promise. Okay. So how do we know if it's a long or short distance question or distance signal? The bottom line is if it was a short distance signal, we would see phototropism having on the right and on the left of this diagram. Because if it was short distance, then those cells at the top of the plant would still be able to send the short distance signal to the cells that are right next to it. And it wouldn't matter that there's this giant impermeable barrier below it. Okay, because phototropism does not occur, that means this can't be a short distance signal. It must be a long distance signal, right? Because the signal needs to travel from the top of the plant to cells that are further down. And so if you actually look at this, the cells that are getting this message are actually going to be on the quote unquote dark side of the plant. So on the side of the plant that is curving. Okay, so if I go back here really quick, this side of the plant that's curving is the side that's getting the signal, right? And because all of these cells need to be signaled from up here, that's going to be a long distance signal and not just short distance things sending it to one another. Okay, so these are the ones that are getting the signal from a hormone called auxin, and I'm going to type that into the chat box in a second. So these cells are actually elongating. They're getting a little bit bigger, which is pushing the top of the plant towards the light. Okay. Again, if it was a short distance signal, we'd still see some sort of phototropism on the right here because all of these cells would still be able to signal to one another if it was short distance. Okay. Next question. Why was the plant with the tip removed and the site? Oh, why were they not phototropic? Okay. So what this whole diagram told scientists, because we didn't know this before, is that whatever it is that's causing phototropism, and now we know that it's cells at the top of the plant that are releasing a hormone called auxin, whatever it is that's happening here, the top of the plant is the part of the plant that is that has these cells in them, these special detection cells. Okay. And the reason we know that is because these are just the results, right? Results are results. When they removed the cap, or when they removed the top, or when they put that thick non-see-through cap on the top, the light was not being detected by the correct cells, and so therefore phototropism did not occur. So this is what they saw, right? And so I think the reason students struggle with this experiment, and this was a free response, I believe, in 2016, which is why I'm spending a good amount of time on it, because when College Board, when AP buys a diagram or creates a diagram or pays someone to make them a diagram, they usually use it more than once, right? So you may see a diagram like this on an AP exam. So stay tuned, right? But you're looking at the results, and we have to analyze what they told scientists. So this is the experiment that was run. They did all of these things. These are the results that they saw. And now we need to figure out why they saw them, okay? And the only logical explanation here is that the top of the plant has to be exposed to light in order for phototropism to occur. Because when it's not, we don't see phototropism, okay? So this just gave scientists the location of the cells necessary to cause phototropism. And then the second experiment is what told them it was a long distance signal, because it only occurred when there was a permeable barrier that allowed the top of the plant, which has the cells in it for detection, to signal the bottom of the plant. Okay, cool, good questions, y'all. It always makes it more interesting when you have questions. To answer Vera's last question about this first diagram, when it talks about slow reversion, it just means that when darkness starts to come, that these things will slowly become inactive. It's not like a funny example here, not like a night and day situation. But basically what's happening is there's a slow decrease in red light and a slow increase in far red light, right? Think about like dusk. Think about like, well, now it's like 4 p.m. when it starts to get darker, which is terrible. Whereas now it's like pitch black. But that slow process is going to slowly inactivate these things, which means that phototropism will slowly stop to occur. Photosynthesis, like the high levels of the light reactions, are going to slowly kind of revert back. That's all that means. Okay, cool. So we covered this, and so I wanted to type in, because this is a really important hormone, and it's oftentimes tested. Auxin is the hormone that is responsible for phototropism. It is obviously going to be light sensitive. And it's going to leads to cell elongation on the quote unquote dark side of the plant, okay, which pushes the plant towards the light. That's evolutionarily advantageous, which is a very large way of saying that this benefits the plant because now it's getting more photons of light, so it is increasing its photosynthetic capabilities, okay? So this is a very important example of how plants respond to light, right? Obviously, plants and light go hand in hand, right? Because photosynthesis is so absolutely crucial. And this is just one way that plants will get more light, and this is the two famous experiments that told scientists how this occurs, okay, which is why it's so important. We're going to practice some multiple choice at the end here that has to do with this process, so stay tuned. All right, another really awesome way that plants use light is called photoperiodism. So this has to do with light and time, right? The period would be like the time, if you think like physics, if you've had physics before, it's the length of time, okay? And this is just another example that you can kind of put in your tool belt of how plants respond to time and light. So one way that plants can do this, if you see on the left here, this is a really cool example. In the middle of the day, so these phytochromes will detect large amounts of light, right? Especially at noon, that's when the sun is going to be the strongest. And plants, some plants, like in this example, I forget what type of plant this is, but I can look it up for you guys. They will actually put their leaves more outward during the day and then kind of tuck them in at night. And that's obviously advantageous. So they're detecting when light levels are high versus low, so that they can maximize their photosynthetic capabilities. They can maximize the amount of chlorophyll molecules that are being exposed to photons, if we want to get really scientific here. But the other really cool thing that sometimes gets thrown in somewhere on an exam is this example over here with short day and long day plants. So basically these phytochromes, they obviously detect light, but different plants are going to respond to the amounts of light differently. Okay, so there are some plants called short day or long night plants that will only bloom when the amount of sunlight in a day is very short. So those phytochromes, again, I told you they were going to come in handy here. They will detect the amount of hours of light, and if it is below 12 hours, right? So like right now when it's just dark all the time, this would be when a short day plant would actually flower. And you can see here, when the day is short, the plant flowers. When the day is long, it does not, right? And so there's some, let me try to find some examples. I'm just gonna pull up a picture for you guys. Actually, chrysanthemum, so mums, if you think about mums, they only come out, do you guys know what I'm talking about when I say mums? Hopefully. They come out all the time in the fall. So if you go to a flower store or flower shop right now, they're going to have mums out everywhere. I have them out on my front patio right they are going to be short day plants. So they actually detect, and that's probably what's in the picture here actually. They're going to detect when the day is really short, and that's when they flower. And that's just the way that they have evolved, which is pretty cool. What you'll see is scientists, they go and mess with these plants. What they can actually do is, what the plants are actually detecting is the amount of darkness, not really the amount of light, which is kind of complex, but they're measuring it in long chunks, right? And so what's happening here is we have this short day, but then scientists mess with the plant and they'll shine a light in the middle of the night, which breaks this up. Now the plant detects this as like 2 short nights, right? Instead of 1 long night. And the long night is what it's looking for to bloom. So now it has 2 short nights, and so it actually won't bloom. So scientists going and messing with things. On the flip side here, and these are again just like really good examples of how plants are going to respond to light, an abiotic signal, are long day plants. Okay, so you can see that here when it has a nice long day, or really what it's detecting is this short night, it will bloom. Most flowers are going to be like this because most flowers are going to bloom in the spring and summer when the days are longer. But that's kind of how they're detecting when to bloom. We can talk about the evolutionary benefit to that in a second. But when they see this long night, they will not. But they will still bloom. So this is kind of the opposite of my third example here. If they see 2 what they would consider short nights, because it's broken up with a flash of light, they will actually still bloom then. So just scientists proving that it's the night, the length of the night that plants are really detecting as opposed to the lengths of the day, okay? Just kind of an interesting thing here. But what I can say, right, going back to these phytochromes and photoperiodism, and we'll talk about germination in a second as well, what's happening here is plants are benefiting themselves by being able to detect how long light is present, right? So if you think about how most of our plants are going to bloom in the spring and summer, that's when days are longer, that's when water is prevalent, right? If you are a plant and the temperatures are warmer, that's when you want to bloom. When the days are long, the sun is out and strong, and there's lots of water available for you to start to grow. So these, over time, right, these plants have evolved ways to kind of detect when conditions are going to be best, which is what you can see here, which is really crazy, but really, really awesome at the same time. All right. So we're going to stop talking about light for a little bit. We're going to kind of move on to something called seed germination. So I know I kind of already mentioned this, but seed germination is this process right here of a seed starting to sprout, okay? And so scientists have done some experiments here too with various chemicals and cell signals, right? And some of the ones are going to be called gibberellins, and then Abscisic. It's my best guess, guys. No, I don't know who really knows how these words are pronounced, but we all just do our best, right? Gibberellins and then abscisic acid, okay, versus an untreated seed, right? So there's going to be a couple things here. So gibberellins we see on the left-hand side, and we'll actually see that that seed is not going to sprout as large as an untreated seed. And basically what gibberellin does for us, I have a whole, I should have grabbed my textbook and put that whole chart of hormones on there, but that's okay. I can do that a little bit later for you guys. But basically, what gibberellin is going to do is it's going to help the plant to, or the seed to digest nutrients, okay? And this would be something that I would be writing down for sure. But it's going to help the seed to digest nutrients and then germinate once the nutrients are readily accessible. And it's also going to lead to a more controlled growth. So you'll see the untreated is actually growing more than the gibberellin seed, but that seed is going to have a more controlled growth. Gibberellin is also oftentimes used and makes larger fruits, right? And so it's kind of like concentrating the plant's energy and nutrients into a more controlled growth that's going to be suitable for the environment, as opposed to just kind of like growing uncontrolled and then not having enough light or nutrients in order to support itself, okay? On the other hand, this is another really interesting hormone to know about, or chemical to know about. Abscisic acid, or ABA, is going to do the exact opposite. Okay, so seeds, if you planted a seed right now, it would not sprout, but it might sprout in the spring, which is kind of awesome, because it's not time. It's not a good time for a plant to sprout right now. If a plant sprouts right now, it's going to die, right? And so what abscisic acid does is it keeps that seed dormant. It keeps that seed nice and protected underneath the soil until conditions are right for germination, okay? So the cool thing about ABA is that it is water soluble. So when there's lots of water around, think March, April, May, right? Lots of water around. This chemical, this acid, will dissolve, and then it's no longer present, and that seed will start to germinate, okay? So now we have this large picture of how incredible seeds are, right? Because they're coated in all of these amazing things that are going to help them to digest nutrients, that are going to keep them from sprouting until there's enough water, and that are going to keep them from sprouting until there's enough sunlight, right? So all of these hormones are coming together to make sure that seeds are only germinating when conditions are perfect for them. If all of these different things did not exist, we probably would not have nearly as many plants on Earth, and we would not be probably surviving at the level that we are. So it's really interesting to think about. So again, I can kind of summarize this for you. Ghrelins are going to be used for growth and development, lead to controlled growth. They help with cell division. They help with digesting, breaking down nutrients in the cell. And then we have ABA is going to inhibit germination, but it's water soluble. So if there is enough water present, more ABA, we'll have germination. Okay, so that's that whole set of things that are going to help the seed to ferment. Okay, only when the time is right, when there's enough nutrients, when there's enough water, when there's enough sunlight. All right, another really cool signal that plants can send to one another is for fruit ripening. So maybe you've heard of ethylene. It's a relatively common actually gas that you might use in like organic chemistry, but it's naturally occurring plant hormone that we most commonly think of in bananas, but it's in all. It's going to be released from all plants, but bananas just are like a really high level of ethylene. And basically what happens is plants can signal one another via this hormone, which is going to be in a gas form, which is kind of interesting to think about. Like if we released our hormones as gases, that would be really crazy. But plants are going to release this hormone in a gas form, and it will signal other plants around it to ripen. And there's a couple of different benefits to that we can talk about. Okay, the number one benefit, and let me just write that for you guys. Number one benefit is if you ever have an unripe fruit, like this always happens to me with avocados or nectarines for some reason, you can put it in a brown paper bag with a banana, like a nice ripe banana, and the ethylene from that banana can actually, come out as a gas and signal to the plant next to it to ripen. And so you'll have a much quicker ripening process than you would if you just kept it on your kitchen counter. So that's a pro tip for you of how to ripen your fruit using AP biology knowledge. But the other thing is, right, we obviously only want fruit ripening at the right time, right? And there's a lot of different reasons for this. The primary reason that plants make fruits is for animals to eat them and then disperse the seeds, aka poop them out elsewhere, right? Because that's the whole point. The fruit is just a protection of the seeds that are inside. And those seeds are meant to be spread so that the plant can continue to grow elsewhere. And the dispersal of those seeds is really important. So it's not just growing right underneath the mama plant, right, but spreading out into the general vicinity nearby. So if plants can have this kind of controlled ripening and signal to one another when it's time to get ripe, then you can have, all this ripe fruit at the perfect time for animals to be eating it and then dispersing that seed, right? It would not make much sense for like an apple or a banana to be ripe in the middle of the winter when a lot of animals might be hibernating or in other areas. And so it makes a whole lot of sense to have this ripening controlled at the right time. For farmers, this is actually used a lot. Someone could write about this. It's kind of like artificial selection, not really, but artificial manipulation, where farmers can actually harvest things early, right? And then they can ship them. So let's say we're shipping avocados from Mexico. We can actually ship a carton of avocados here that are unripe. And then when they get here and they're ready to go to grocery stores, they can be actually sprayed with ethylene and they'll ripen up to the appropriate amount for consumption. It's pretty crazy. But yeah, ethylene is a really important hormone to know about and kind of why it's important. And then from an evolutionary standpoint, it's going to help with seed dispersal for sure for plants. which is really, really important for their continued survival. Okay. All right, I'm trying to think of how many. I just have one more. Well, sorry, I'm flipping through these. What the heck? One more overall example for you, but all these are really, really important to just keep in your brain. So it might be important to just like go back through your slides or rewatch this and just hear all the different examples of how plants are responding to signals. Maybe go hug a plant today, thank a plant for all it does, right? Because it's pretty crazy. But the last thing that we'll talk about briefly is this idea that plants are able to respond to gravity. And there are a lot of different things that go beyond the scope of the AP course, so you don't need to worry about them, that contribute to this reaction. But it's important to know that plants can actually respond to gravity. It's called gravitropism, moving towards or against gravity. So you'll see we have negative gravitropism, which is listed here as geotropism, which is the same thing. But negative gravitropism means it's going to move away from gravity, right? Which means that the part of the plant that needs to be up by the light is going to move up by the light no matter what. And the part that needs to go down into the ground, the roots are going to go down into the ground, positive gravitropism, 10 times out of 10. We could flip that seed and it will still flip its roots and its leaves, right? So let me see if I can grab another picture for you guys. I love gravitropism. I think it's so cool because this is obviously, I like this picture. The reason I picked it is because a plant can tip over, right? It's started to grow like this with the leaves up and the roots down, and it can tip over and it will actually respond, right? It's not just going to keep growing in that direction. It will actually respond, and you'll see the plant start curving up like it's supposed to, and the roots start curving down. And then if you switched it again, it would correct itself again, which is really, really interesting. But you see. Here we go. This is a really good example. Let me just copy it in really quick. Should have added this earlier because it's like flipping a developing seed, because seeds are going to be able to respond and know which way's up and which way's down, which is why you don't have roots popping up out of the soil and leaves popping down into the soil. But what you can see here is this guy obviously was starting to grow leaves up and roots down. And then we flipped it. So we have the leaves down here and the roots up here, and it's able to still kind of curve around and grow up towards the light and down towards the ground, which is just like an amazing evolutionary benefit for this guy. Plants are like just a wonderful, immense version of all sorts of evolutionary triumphs, which is really pretty cool. So I really like talking about abiotic signaling for a couple of reasons. One, it highlights cell signaling. Two, it highlights many, many evolutionary things. And then three, it just gives you appreciation for plants, and we should love them because they're really awesome. So what I can say about plants is they don't show up on the AP exam usually in isolation, which is why I like to teach them only through photosynthesis and abiotic signaling, because those things have larger implications, right? Like chemical implications and cell signaling implications that are going to be really, really important connections. And that's why they are focused on in the AP exam. And that's how they are focused on in the AP exam. But they're never going to ask you a question about a plant, like about its leaves or its stems or its internal organs or anything, or like, you know, sexual reproduction stuff. They're never going to ask you about that. So it's usually just in tandem with stuff like this. So I have a couple practice problems for you that hopefully we can get through. And the best way to do these, and I know I say this frequently, but you really just need to kind of take some time, read through it. I'm going to actually have you guys work on all three that go along with this graph. So I want you to look through the graph. You can kind of flip back and forth on your own time. Read through the three questions. We're going to spend maybe like 4 minutes trying to get through all of them, and then we'll bring it back and we'll chat about the right answers and why they're the right answers. There's some tricky words in here to look out for, so just make sure that, and I'll give you some pitfalls, but these are very classic AP style questions, so make sure you're reading carefully. Okay, so I'll give you about 4 minutes to look at the graph, read through all three questions that follow, and then stay tuned for some answers. And then I'll answer all of your questions too when we come back. But I want to make sure everyone has time to just look through these questions for a second. 30 more seconds. Okay. Hopefully that was enough time. Was it? Someone tell me if you're still working. Yeah, you can just yell at me. All right. We're just keeping moving. Okay, so let's check. So according to the data, at day 8 of the experiment, all the following statements are correct except. So I don't know if your teachers do this. It's technically not a good way to write a question to have except in it because it's overly confusing, but guess what? The AP exam does it all the time, okay? So you have to look out for these questions. Sometimes our eyes are selectively blind to them. Like we were doing this in class the other day. I hadn't seen it in a little while. It'd been like a year, right? And I was like, oh my God, the student was explaining the answer and he was 100% correct. And I was like, oh my gosh, I was not paying attention to the word except. So it happens to the best of us. As you're looking through these, hopefully you realize that the answer choice was D, okay? And the reason being, at day 8, and that's all we're looking at, T is actually lower than DG. And so it is not plausible that we can predict that T will eventually be taller if we're just looking at day 8, okay? If you work through the rest of these, T, DG, DGA, seedlings are all the same height. That's very true, or similar height. D seedlings are less than half, also true. And T, DG, and DGA seedlings appear to be in a more rapid growth phase, also true because their slopes are higher, right? That's how you would determine like a rate, which would be the rapid growth, than D, okay? So your answer there is going to be D. The eventual greater height over the DG seedlings is already predictable. 2, a hypothesis these data, which is actually correct, data can be plural and singular, tend to support would be that what? Great question, okay? So here's where we have to be looking, and 2 and 3 are going to be very similar in this way. We have to be looking just at the data that is given to us, okay? So the answer here is actually A. A lot of my students seem to like to pick B, okay? C and D we can eliminate. So Auxin is not going to promote cell division, but cell elongation. It's just a technicality, but there are better answer choices. So D is not going to be it for us. And dominance, that is something that we have not even talked about yet, and it's certainly not highlighted on this table. Just because something is taller doesn't mean it's dominant. So that's an important misconception to get out of the way. That's why we can eliminate C, okay? The reason A is the best answer choice here as opposed to B is because this guy just has a dwarf seed, and this guy has the dwarf seed plus gibberellin, and you'll see it is remarkably taller. Okay, so the evidence is showing us that with gibberellin, this guy can grow much, much taller. The reason why B is not correct is, and this is going to go into question 3 as well, when we have gibberellin and auxin, the plant is actually lower than when we have just gibberellin. So if gibberellin and auxin were both contributing to cell elongation, then we would see a plant that is way taller here when both of these things were in action. Okay, so something is happening when gibberellin and auxin are present that's actually decreasing this. So they're not working together in cell elongation, right? Otherwise, we would see a plant that's way taller. And so the only one that's logically represented by data is A. 3, I kind of already gave you the answer, but I really like this question because sometimes students just like to extrapolate and come up with these crazy ideas. And they're like, oh, well, D seedlings are shorter, so maybe they won't produce new seeds and mature, right? Maybe that's true, but it's certainly not shown on the table, okay? So we just have to look, or in the graph, we just have to look at what is provided to us. That's the only thing that we can use to make any sort of conclusion here. And A, C, and D all represent things that are not shown in the graph. A is talking about matureness and seed production. C is talking about stem strength. And D is talking about internodes. None of that is talked about. But we do know that auxin partially counteracts the action of gibberellin for the exact reason that I just said. Gibberellin, really, really high. Gibberellin and auxin, lower. So something is being counteracted here that's decreasing the eventual height of this plant. And that is what we are talking about. And that's the only thing that we can prove with data. Okay? All right. I'm going to start answering a few more of your questions then. So Vera, as far as C4 and C3 plants go, usually C3 plants are the ones that are going to be those flowering plants that we're talking about, but these things can actually exist independently of short and long day plants can actually exist independently of C4 and C3. So there's no like direct correlation there because both can kind of be either. Oh, and as far as other AP multiple choice questions, I certainly can. There are practice tests that you guys should definitely download if you can that are released each year. Unfortunately, so these questions aren't from an actual AP test because I can't release those. It's against the law. But I have a couple other resources of AP-like questions that I would love to share with you guys. Oh, and all of the free response questions are free games. So you can look all those up on your own time. You just want to look from 2013 to 2018 because 2013 is when the redesign happened. So 2013, 14, 15, 16, 17, and 18 are all really good resources, and all of those are free for you to use. You just have to search AP Biology free response, and then you can put in the year. All right, for gravitropism, Salam. Yes, leaves are always going to be negative because they're going against gravity. That's why we call it negative gravitropism because they're going against gravity. That would be a negative reaction. The roots are always going to be positive gravitropism because they're going with gravity, right? They're going down while leaves are going up. That will always happen because that's the evolutionary advantage for the plant. All right. We're going to skip the short and long plant one, but let's just take a look at this last one. So this is going to be looking at the exact same image. So this should be pretty quick and easy then that we were talking about earlier with phototropism, but there's two application questions that follow. So we'll give you about a minute or two to look through them and make your best answer choice. And then we'll talk about why, and we'll call it a day. Okay, so this top question here is really important because it's a very challenging question. So let's see how we did. So you need to be very familiar with this experiment in order to get this question right, which is why I like to showcase it to students. So what you need to know is you need to know that through this experiment, we already know what the stimulus is for the bending of the plant. It's the light. And we already know where the growth factor is being made. It's at the top of the plant. Okay, so we can eliminate A and D, even though they're very logical, even though they have everything to do with this experiment. It's not a next logical step because we already know the answer to that question. Okay, so you can eliminate A and D. Between B and C, determine which side of the tip is making the growth factor or determine what is inhibiting mitosis on the dark side of the plant. We have no reason to believe that mitosis is being inhibited. In fact, when we talked about the dark side of the plant is actually going to have cell elongation. Okay, so it's not that the cells are not capable of dividing. So that's kind of a stretch. But the only then solid next step would be determining which side of the tip is making the growth factor. So the best answer choice here is B. Not that C is a terrible choice, but B is just a better one. And you have to become really comfortable, just like on the SAT, with knowing that there are good and great answer choices. And B is going to be a better answer choice than C, just because, again, we have no reason to think that mitosis is being inhibited here. Okay, so it's just taking it a step too far. Next question, which of the following best represents the results of the experiment? Okay, so this goes back to talking about which side actually has the growth factor on it, and I already explained that this is going to be the dark side, so hopefully you were able to kind of get that. So C and D, hopefully you were able to eliminate because the agar block is not going to have an increase in height necessarily. It's just moving towards the light. The agar block is not also going to cause the coleoptile to bend in either direction. It's going to be bending towards the light. So the coleoptile, which is the little plant, is going to bend away from the side with the increased concentration of growth factor, right? So that's the dark side of the plant. So those cells are going to expand and it's going to push it towards the light. Okay, that's a really tricky application question that kind of goes off of what I was talking about earlier. So you can always go back and rewatch that. But remember, the dark side is actually what has the Auxin reaction that's pushing it towards, right? Those are the cells that are getting bigger. If these cells were getting bigger, it would push back, right? So these cells are getting bigger, so it's pushing towards the light. So that's just an application of knowing kind of what's going on there. But the coleoptile is going to be bending away from the side with the increased growth factor. So the answer there is A. Okay, very kind of a tricky application question. Looks like we have one more question. Oh, the coleoptile is just the little plant shoot. That's just the name of it. So that's what they're talking about there. So this guy is the coleoptile. Okay, and it's moving away from where the growth factor is because it's moving towards the light, which is the stimulus. All right. So this last question is just about short day plants. So short day plants only flower when days are less than 12 hours long. And so you'll see that there are two examples of where we have, we have two examples. Yep, example B and C both have a day that is less than 12 hours long. But in example B, an 8-hour day and a 16-hour night with a flash of white light after 8 hours, that night is not going to be long enough, right? If you go back and look at the example, they're really counting that long night. And so having the 13-hour night and then the 11-hour day with a minute of darkness is not going to affect it at all because it's only going to flower when the days are less than 12 hours long as is. So if anything, it's going to make it flower more. So if you read through that example, you'll see why C is the best answer choice. So that's all I have for tonight. Next week's a big one. We are going to be talking about DNA and DNA replication. So we're going to move kind of out of this cells unit and into more DNA kind of stuff and genes and proteins. And we're going to talk soon. We'll talk about transcription translation, talk about gene regulation, which is overly confusing, but we'll get to the bottom of it and all that fun stuff. So definitely stay tuned. Bring your friends. Talking about DNA is super fun and exciting, and it also shows up all the time on the AP exam. Super important topics. Rewatch this if you have any questions or reach out. But otherwise, enjoy your night and I'll see you next week.